This conference will now be recorded.
Okay, so we have just a little tiny little bit here to finish up talking about blood. And then we are going to move on, to the next part of this unit here, which is going to be about the heart. Now, the last of our formed elements here to discuss, right, are the platelets, thrombocytes, technical term for it. And again, what's going to happen is as we make them, and we'll go through this process in the next couple of slides, is literally we're just going to have a cell kind of explode. Right? And the little tiny little bits of cell membrane that are left over from that are what actually make the thrombocytes themselves. Typical sort of normal numbers to expect if doing a CBC, right? Somewhere around maybe just south of, there we go, just south of maybe 500,000 to maybe 150,000 per microliter. Pretty average number there. And you can see there's quite a range in there. And that range is due to whether or not you had to recently undergo a clotting event. Right? So you will see some variation in there, but anywhere in that range is going to be perfectly normal. Now again, when we talk about what platelets do, right, really they're kind of that middle step in vessel repair, right? We have the spasm to start us off, we have the coagulation to finish us off, but the middle part here is sort of the emergency patch kind of thing. This is the platelet plug, really, that forms in there. You'll recall that we had some discussion about ineffectual treatments for sickle cell anemia there, the inhalation of nitric oxide. What nitric oxide is really doing is more or less just inhibiting platelet plug formation. It's part of what keeps them from sticking to each other when we don't want them to. Okay. Now, because they're just chunks of cell, detritus really, they don't have a long lifespan. We're looking at about maybe 10 days here, kind of at the outside. Right? So again, fairly regular, fairly rapid turnover of these things, because, like I said, they don't last particularly long. Now, as far as the process goes, we're going to have thrombopoietin, right? So we had erythropoietin and leukopoietin. Here we have thrombopoietin starting us off. And the process here is really, 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 really simple. Okay. We're going to start, right, from the hematopoietic stem cell. Okay. And the pathway then is just this cell undergoing mitosis after mitosis after mitosis after mitosis, right? But not cytokinesis. Okay, so if you remember back in ANP1, right, we talked about those two as related but separate processes, right? Mitosis being the replication of DNA and the division of the nucleus, right? And cytokinesis being the division of the cell into two daughter cells. Well, what happens here is we continue to kind of replicate the nucleus, right? We replicate that DNA and we fill and fill and fill. This is what mega karyoblast means. Karyo here meaning meaning DNA, right? Mega meaning a lot, right? And blast, of course, meaning cell. So we're just going to kind of fill this cell up with the products of mitosis, right? And just like a balloon, as you continue to blow up a balloon, eventually it's going to burst. Okay, and that's ultimately really what happens here. Right? We just fill the cell up until the membrane can't handle it anymore and it ruptures. And we're left with little tiny little fragments here that end up being our thrombocytes, that end up being the platelets themselves. So it's a significantly simpler process really than a lot of these other ones that we've gone through. Okay. So as we've seen through my mishap here at the computer, next part here for us is going to be what's going to be moving this blood around the body, right? The heart. The heart in and of itself is an interesting thing in that it is kind of two separate pumps that are kind of just coalesced into one solid unit. We have one pump that pumps blood through what we'll call the systemic circuit. Okay, that's going to take oxygenated blood out to the body and return it back to the heart. And then we've got a pulmonary circuit from the left side, where we're going to send blood out to, or rather the right side, excuse me, out to the lungs and back. Okay. So the right side is going to be receiving blood from the body, sending it to the lungs. The left side receives it from the lungs, sends it to the body. So you can kind of think about the pathway of blood as more or less being a big figure 8, where you kind of have a crossover. That little crossover event happens at the heart itself. And that's going to connect our two different little circuits. The heart can also be thought of as being upper and lower parts. Okay. And so we think about the upper parts. These are going to be the parts that receive the blood, either from the lungs on the right side or from the lungs on the left side, the body on the right side. It's been a long morning already, sorry. And we call these the atria, right? And an atrium, if you've ever been to, a hotel, right, or anything like that, right, you'll walk into a lobby or an atrium, right? It's sort of the receiving place. It's where you enter, okay? And so our atria are the chambers that receive blood, or where blood enters the heart. And again, we're going to kind of revisit this right versus left pathway quite a bit. So just kind of bear that in mind. And then from there, we're going to go lower chambers of the heart. Again, both the right and the left side. And here, these are the ventricles. And these are going to be the muscular portions of the heart, the ones that are responsible for pumping blood out to various parts of the body, either via the systemic circuit, left side, the pulmonary circuit, on the right. So in the Mary Ibn Hone book, and also in the OpenStax book, if you have that one instead, there is a figure much like this one that kind of takes a relatively simplistic look, I think, but is a pretty good kind of thought on the pathway of blood through these two different circuits, and again, how they cross over here at the heart. Now, I'm just going to remind you here that although convention is almost invariably for deoxygenated blood to be blue and oxygenated blood to be red, of course, recall that all blood is red, just varying shades thereof. The heart itself, if you were to make a fist, firmly plant your elbow at your side and cross over to the midline of your chest, that's roughly the size and position of it. It's not all the way over here. A lot of people think it's all the way on the left side. It's actually a midline structure. It just so happens that the ventricles are rotated slightly in such a way that the sort of the apex of the point of your heart is kind of on that left side. But it is, again, right down the middle. And then therefore it exists within the mediastinum, that middle cavity within the thoracic cavity, the space between the lungs, basically, where things like your esophagus and your trachea and your aorta and your vena cava and all those sorts of major vessels and pathways also reside. So it's basically all the stuff kind of in the dead center of your thoracic cavity. It sits right on top of and actually depresses the diaphragm slightly, so there's a little part that sits right in there. And comes up pretty high, right up, I would say, up until sort of the second intercostal space, and so between the second and third, sometimes as high as the first intercostal space. So it's a pretty large structure, right, overall, but again, it's a fist size. As far as the terminology here for some of the basic landmarks of the heart, it's a little bit confusing, okay, in terms of what the names of these things are until you learn about the developmental anatomy. And basically the way that these things develop is that the heart starts out life as basically two muscular tubes side by side, that then kind of rotate and flip around. So what embryologically is the bottom becomes the top, and what embryologically is the top becomes the bottom. And so the terms base and apex kind of have an opposite meaning to what you might expect, in that the base of the heart is at the top, and the apex is all the way at the bottom, sitting on the diaphragm. So bear that in mind as you kind of remember some of these terms. And in fact, you can actually get a sense for precisely where the apex is. Don't do this now. Do this at home in the privacy of your own space. But if you feel your chest just kind of to the side of your left nipple there, you can usually feel a pulse there. That's called the apical impulse. That's right about where the apex of the heart actually sits. And you can actually feel that through the lung and through the muscle of your chest. So if we look again, kind of where we're at, right? Again, sometimes this surprises people, right? But it is right on top of the midline. It sits right in the middle here. The only part that's really off to one side or the other are the ventricles, and really just the apical part of the ventricles down here, the apex of the heart. So again, typically in the second intercostal space, sometimes all the way up here to the second rib, but really the second intercostal space down to the fifth intercostal space. If we take a slightly different look at the anatomy here, we'll kind of cut through roughly around here, right around T7. Here is a transverse section through the thorax, right, with our lungs, left lung, right lung. We have the mediastinum here in the middle. Okay, so you can see here the esophagus, the descending aorta, the inferior vena cava, vessels either leaving or coming to the heart, and the heart sitting in this space right here. If we look at it kind of a little more in situ, we've removed ribs here, the bony elements here, but we've retained the lungs in place. Again, you can kind of see the close relationship between those two. In order to really see all of the heart, we have to, as you can get a sense for this now, but reflect the superior lobe of both right and left lung to really see it. Now, something that we're going to be talking about again and again and again and again and again whenever we talk about any organ is that, generally speaking, we've got multiple layers of connective tissue that surround each of these organs. Okay. And typically what we're going to find is that there's going to be a layer that sits directly on the organ and a layer that sits on the body cavity. Okay, so in this case, the mediastinum. And then the space between them, there's typically also going to be fluid. Now that fluid is almost invariably always going to serve the same purpose, lubrication. Your organs are at best loosely attached to part of your body, and so they have a lot of wiggle room. Right? And your heart, of course, is beating 60 to 100 times a minute at rest, right, within this space. So it is moving on its own as well here. And so the risk that we'd run would always be kind of wear and tear as we rub on and move against other organs. And so we have this kind of repeated anatomy of surrounding these tissues in a fluid-filled sac, basically. The outermost layer here with the heart, though, is actually going to be outside of that, and this is going to be the fibrous pericardium. And this is going to be a little bit unique. Most organ systems don't have an outer fibrous layer in the same way. But most other organs also don't have this sort of filling expansion that we see with the heart. And so we have a fibrous outer layer here that's going to kind of serve as a upper limit on the expansibility of the heart. Right? It's going to be largely collagen, and so it doesn't stretch. Right? So the heart can only get so full. This is relatively important. Inside that fibrous pericardium, okay, pericardium here meaning just around the heart, right? We have a more typical membrane here, a serous membrane called the serous pericardium. And this is going to be what's going to be containing that fluid, in this case, pericardial fluid. We like to keep names as consistent as we possibly can. Now, with the pericardium or the pleurae or any of the various membrane systems that we've got with our internal organs. Again, like I said, there's always going to be a layer that's going to be against the body wall and always going to be a layer that sits on the organ itself. Okay. The terminology here is going to be the same regardless of which membrane it is. So for the pericardium, we have a parietal pericardium, which is going to be on the body wall, and a visceral pericardium, which is going to be on the heart itself. Now, these aren't two separate membranes. That's actually one membrane that kind of folds over at the edges and kind of starts on the heart, kind of has a little fold and goes out to the organ, but we talk about them separately. And they together define this pericardial cavity, which is of course filled with pericardial fluid. We're going to talk a little bit more about this in a second here. The outer layer of the heart is called the epicardium. It's actually the same thing as the visceral pericardium. So as we start talking about that, we'll revisit that at that time, just so you're aware. So if we look at what's going on here, so here we've got the heart muscle itself. Here is the pulmonary trunk, so this is going out to the lungs. We've got this outer fibrous layer, right, the fibrous pericardium, and then the more typical membrane system here, the serous membrane, within that. So the parietal layer sitting on that fibrous pericardium, making a little loop here and going on to the heart itself, being the visceral layer there, or of course the epicardium. And then the space in between pericardial cavity, then the pericardial fluid. Typically, you will see, not infrequently, depending on what kind of practice you're in, the sort of swelling of the pericardium, right? Pericarditis, right? Again, of course, remember, itis simply means inflammation, right? So inflammation of the pericardium, not terribly uncommon, not going to necessarily kill you, but it does induce something called cardiac tamponade, which is not pleasant, I guess we'll say that. What's going to happen here, remember this fibrous pericardium is non-expansible, right? It's giving you an outer limit, right? And so everything within that sac kind of has a maximum volume. Okay? Well, with pericarditis, what we're doing is we're dumping a bunch of fluid, a bunch of extra fluid, into that pericardial cavity. And that's going to be compressing the heart. I suppose if it was severe enough, it could limit the ability to pump blood altogether. But generally speaking, it just kind of is something that really only shows up when someone engages in any kind of non-rest activity. It can be bad enough that a person has trouble walking across a room. That's kind of a severe incidence of pericarditis. But typically, tamponade is just unpleasant, but leading to kind of more severe outcomes eventually. You'll be able to diagnose this fairly easily. When you go in and you do your 4 heart sounds, what you'll hear is this kind of, hard to describe. It's kind of like a cross between like a wood frog and one of those haunted house door, creaky door sound effects. Cardiac friction rub is what it's called. And it's a really, really characteristic sound. It's not mistakable for anything else. But that's going to be your primary indication of pericarditis. As far as the heart itself goes, we're going to have a 3-layered structure here, but one of our layers, again, we've already discussed. All right, so we have the outer layer, the epicardium, okay, which again is the same thing. It's literally the same tissue as that visceral pericardium. Then we have a muscular layer in the middle, the myocardium. Then we have an inner layer, the endocardium. So, like I said, we're not going to really spend much more time with the epicardium. It is what it is. The myocardium, on the other hand, is probably what you think of when you think of the heart. It's where all of the cardiac muscle is. Um, and the arrangement of muscle in the heart is really pretty interesting. It's not sort of these maybe sort of round sacks of muscle, maybe sometimes people think they are. They're actually arranged in this really intricate spiral pattern with little tiny little bits of cartilage serving as anchors, connective tissue serving as anchors in the middle there, forming what we call the cardiac skeleton, which is also, of course, a part of this myocardial layer. Come back and we'll talk about the cardiac skeleton again. Also, when we talk about electrical signal transduction through the heart, because all of the wiring that connects the various regions of the heart electrically also resides in this myocardium. And then the endocardium is, to me, pretty interesting. It's a relatively thin epithelial layer, right? Because it's a lining, it's epithelial. And its job is to be as frictionless as possible. Right? We would like to limit friction or drag or any of these sort of regressive forces as much as possible. But to me, the fascinating part about this is that when blood vessels leave the heart, the pulmonary trunk, the aorta, right, or they come back to the heart as the vena cava, right, or any of these things, the lining of those blood vessels and the lining, in fact, of every blood vessel in your body is continuous with the endothelium, or the endocardium here. It's one continuous layer of tissue that is basically the part that touches blood all around your body, which is pretty wild for me to think about. So we go back to our little cross section here, right? We had before our fibrous, parietal, and visceral pericardium. It's of course also the epicardium, the myocardium, the muscle part here, and the endocardium lining, all of that. If we look at, and this isn't all of the cardiac muscle, but this is just a handful of the cardiac muscle out here, just to kind of, again, show you what I'm talking about with this kind of elaborate spiraling pattern. This is kind of what we would expect to find. It's really almost artistic in a way. Okay. So again, as we move inside the heart, right, this is where our chambers reside, right? Our two atria, our two ventricles, upper and lower, right and left. And one of the really, 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 really important things for us and all other mammals energetically is that we keep oxygenated blood and deoxygenated blood as separate as possible, right? So we are not going to have any crossing over, right, from the right side of the heart to the left side of the heart. We want to keep those things completely separate. And so keeping those sides separate are two different septi. And we have a atrial septum and a ventricular septum that are complete. They block flow from one side to the other. Now, this is not always the case. In utero, because we are not using our lungs, we have no need whatsoever for a separate pulmonary circuit. In fact, it's a wasted circuit for us because our lungs aren't filled with air. They're actually filled with amniotic fluid. And so what we're going to see is that the interatrial septum in utero actually has a fairly large hole in it, okay, called the foramen ovale. And that's going to connect the pulmonary and systemic circuits in utero. So the blood just kind of continuously circulates without being separated. As we get close to hatching, right, and ideally by the time we hatch, although it does sometimes take a week or two post-birth, postpartum, to actually close up, that hole, that foramen ovale, does in fact close, and we have a little scar left on that, the fossa ovalis. It is most prominent on the right side, but you can see it regardless. The interatrial septum is always solid. There is never supposed to be any gaps or any holes in that separating those ventricles. So if we look at a cartoon, here of what's going on, right? We have our atria here, right side, left side, our ventricles below, right and left. Right? You can see clearly here the interventricular septum. Right? The interatrial septum is obscured a little bit here by this blood vessel here, the pulmonary trunk, but you can see kind of a little bit of it here poking up behind with that little fossa ovalis there on that right side of the interatrial wall. If we look kind of again at some more gross anatomical landmarks on the heart, we've got some fairly easy to find things. There are a series of grooves on the outside surface, one that circles the entire heart from the outside and basically serves as the separation, the external indication, right, of the separation from the atria and the ventricles below. This is the coronary sulcus, or the atrioventricular groove. That also is a term that we can use. And again, when we look at the heart from the outside, this is kind of where we know atria, ventricles. It's also going to serve as a pathway for some fairly important blood vessels in coronary circulation. So as we talk about how the heart itself gets blood to its muscle, we're going to come back and revisit the coronary sulcus here. And then on the anterior and posterior sides of the ventral part of the heart, we have the anterior and posterior interventricular sulci. And these mark out where the interventricular septum is. Again, it's kind of our landmark for it on the outside. So again, thinking about it as kind of, this is the right side and that's going to be the left side. Again, like the coronary sulcus, there are going to be some relatively important blood vessels that travel in this, or in these sulci. So again, we'll come back and we'll talk about those at that point. Continuing a little bit with sort of external anatomy here, when we look at the atria, kind of functionally, they do have muscle associated with them. Okay, they do have a myocardium, right? But not a lot of muscle. The atria really don't squeeze, kind of in the same sense that the ventricles really do. And the job of the atria really, honestly, is only to fill up with blood. Okay, and so it's not elastic tissue, it's not stretchy tissue, right? And so what we're going to need to have then kind of is sort of a bagginess, right? So that when we are full, we can kind of expand it and fill that all up. When empty, right, what's going to happen then is the atria kind of sag, and we're left with these sort of dog ear kind of structures, right, called auricles. An auricle, term you might remember from when we talked about the ear, right, because it's literally Latin for ear. And my dog Baxter is a German Shepherd, right, but he's got some beagle in him. The only beagle in him that I can tell is his ears don't stand up like they're supposed to, right? They just kind of flop down to the side. That's kind of what these look like. They're just kind of floppy little structures off to the side that give expansion room for the atria to fill up. On the inside of the right and left atria, again, there is some muscle here. Largely the passage of blood out of the atria into the ventricles is mostly driven under gravity rather than contraction of muscle here. But there is some muscle, not a ton, but some, largely in the form of, I'll find my cursor again, these things called pectinate muscles. Now the right side has more of this than the left side, but really neither side is particularly muscly. And in fact, when you get in and start looking at these structures, the walls are deceptively thin. There's just really not a lot to them. Again, just because they don't need to. They don't do any real work other than filling with blood. They're kind of a, like I said, like a lobby for a hotel, right? They just a place where you come in and move on to your destination from there. Right? So like I said, they're really not going to do a lot of squeezing. What they do though, right, again, is receive blood. And so the major vessels that return to the heart all come back to these atria. So on the right side, right, coming back from the body, okay, so bringing deoxygenated blood back, we're going to have three big vessels that come in here. We have the superior and inferior vena cava. Okay, superior comes down from the head and the upper limb. Vena cava comes, or the inferior vena cava comes up from below, so the abdomen, the lower appendages, all that kind of stuff brings back in there. And then we're also going to have a little kind of sinus almost, the coronary sinus on the back side of the heart, which is going to be a collection area for blood returning from the heart muscle itself. So we talk about coronary circulation, delivery to the heart and from the heart. Coronary sinus is where blood comes back from the heart itself. On the left side, we're going to have four pulmonary veins coming back from the heart, two on the right and two on the left. They don't get special names, they're just pulmonary veins. And from there, right, as we drain into these atria, either pulmonary veins, vena cava, or coronary sinus, we then fill. That filling drives blood through a couple of valves and down into the ventricles themselves. These are the largest part of the heart. They're a significant chunk of the volume. Not because they hold more blood than the atria, they hold exactly as much blood as the atria do, right? But because they are so much thicker in terms of the muscle, right? The myocardium in these things is really substantial. Okay. And that substantial increase in muscle leads to a fairly robust increase in volume of these structures as a whole. Now, one of the curious things that's happening here, and you'll see this when you look at the position of the interventricular sulci, is that while the right atrium is on the right side and the left atrium is on the left side, the ventricles are twisted slightly, such that the right ventricle is actually kind of in front. So it kind of does this sort of clockwise or counterclockwise twisting. Right ventricle is mostly in front and the left ventricle is mostly behind. Again, there are some spacing reasons for this in terms of getting blood vessels out. They kind of have to twist a little bit to make that work. But they're not arranged in the same way that we see with the atria. Because they are significantly more muscly, they are major propulsion unit of the blood. The inside of these things is also significantly more marked by muscle. In fact, when we look at the inner surface of these guys, what we're going to see is there's all these little sort of ridges or kind of like strings, kind of looks like Freddy Krueger's face kind of a thing. These things called tuberculae carnei, which is literally Latin for meat walls, or meat columns, I guess is a better way to put it. Meat ridges, maybe. Just because of the sheer volume of muscle in there, it actually kind of expresses itself as these little grooves on here. We're also going to have these muscles here called papillary muscles. These are going to be attached to the flaps of the valves, and they're going to hold those valves shut when the ventricles contract. So they don't open them. A lot of people think that they open them. They don't. They hold the valve shut under contraction. And we'll talk a little bit more about that as we talk more about the valves in a little bit. Now, because we are pumping blood out, we have, maybe it's not obvious. One of the important things about this is that we leave via only a single vessel. Okay. So the right side has one vessel that leaves, the left side has one vessel that leaves. Rather than having multiple vessels coming in, we have one vessel going out. This relates in large regard to the generation of blood pressure and all these other things, but it's physiologically important. The right side, pumping blood out to the pulmonary circuit, pumps blood out into the pulmonary trunk right up front, right? Because again, remember the right ventricle is on the front side of your heart. And then the blood leaves the heart from the left side via the aorta. The aorta is the single largest blood vessel, as far as I'm aware. Certainly the single largest artery in your body. So looking at some of this anatomy that we've been talking about, here we have the atria above as delimited here by this coronary sulcus going around the heart. These sort of flappy dog ear structures on the outside here and here. These, of course, are the auricles of those atria. One full kind of swell and expand. Here we have the anterior interventricular sulcus. If you flip the heart around, you'd see the posterior one. And coming out from the right side, so here is the right ventricle here, mostly in front, the left one mostly behind. Coming out, we have pulmonary trunk, then splits off to go to the right and left lungs. Coming out of the left ventricle from behind, then up and out here through the aorta there. And here on a real heart, maybe a little easier to see the floppiness here of the atria, right? These auricles, a little more obvious than a sort of a dead heart here. The right ventricle, the left ventricle, the interventricular sulcus, coronary sulcus. One of the tricky parts about this is that, of course, there's a lot of fat on the heart in general, but also once you get into a real animal, nothing is color coded, right? So identifying things like the pulmonary trunk, which is here, coming out, branching out this way, right, or the aorta, which is actually covered in mostly fat at this point, but coming up and coming that way. Once we look inside, here, getting a view of the inside of the right ventricle in this case, you can see these trabeculae carnei, right? These sort of neat ridges. Okay? And then here, attaching to the flaps of the valve, we have these structures called chordae tendineae. We'll talk more about those when we talk about valves. But the papillary muscles here and there attaching to those chordae, holding them in place during closure. Valves themselves are significant. When we talk about the circulatory system as a whole, we're going to see lots and lots and lots of valves because just like with the nervous system, right, we want single direction flow. If we start flowing backwards, that defeats the whole point, right? It is at best extra work because we have to pump the same blood more than once. It is at worst going to result in hypoxia and maybe death, right? So we want these valves in place so that you can only go back so far, right? We limit the sort of regression possible. So we see them a lot, for example, in major veins in the body, not so much in arteries, but in major veins, and then separating out the chambers of the heart and also sort of serving as a gate between the heart itself and the major vessels leaving it. So we have two valves here that we're going to refer to as AV or atrioventricular valves, one on each side. And these are going to govern the flow of blood between the atrium and the ventricle below it. The right side is called the tricuspid valve, okay, because it has three flaps, right, or three cusps if you prefer. And the left side is the mitral valve or the bicuspid valve because it has two flaps. There's not really any particular reason that there's two versus three, they just are. Right? One's not stronger than the other or weaker than the other, they just kind of are. And again, their whole job, right, is that when the ventricles contract, what does it do? It puts pressure on the blood inside of it. It squeezes the blood inside of it. We want to make sure that that blood goes up and out and not back towards the atria. And so we have these valves that slam shut during ventricular contraction to keep blood on that side. This is why we have the chordae tendineae. This is why we have the papillary muscles because when we're trying to keep them shut, there's a lot of pressure trying to make them fail and flap open backwards. Okay, and so we have these additional little structures there that keep them closed. And if we look at a cartoon here of what's going on, there are some bits of anatomy here that help them in this role. So the valves themselves, right, are not flat. They're actually curved. And the curve, the concavity is facing, in this case, down towards the ventricle. And the reason for that is that when we contract that ventricle, right, blood is going to want to squeeze and go out, right, and it's going to go up. And part of where it's going to go up is against these valves. As it fills this space, then it takes these two valves and it pushes them closed like this, right? Because they're concave down, they get kind of slammed together and pushed against that. Now, with these valves, because we have so much pressure being induced, right, there is enough pressure here to get blood from your heart all the way down to your big toe and back, right? So there's a lot of pressure here. It's hard to resist all that. And so that's why in this case, we have this additional little safeguard here, these chordae tendineae and the papillary muscles that, again, anchor just the little tips here of those valves, and they keep them from everting, they keep them from flapping backwards and failing. The other set of valves that we have, we call these semilunar valves, and these are between the ventricles and the blood vessel that leaves them. So there's one between the right ventricle and the pulmonary trunk, and there's one between the left ventricle and the aorta. They are conveniently enough called the aortic and pulmonary semilunar valves. And the job of these valves is to prevent blood from flowing back into the ventricles. So just as the atrioventricular valves kept blood from flowing back into the atria, here they're going to prevent them from flowing back into the ventricles. Now, the general anatomy here is more or less the same, right? We have curved valves, okay? Only because the direction of flow, or the direction we're going to try to prevent, I guess, is 180 degrees, right? The valves themselves are turned 180 degrees. So instead of being concave down, they're concave up, because what we're trying to prevent is blood from flowing from here back down. Right? Now, we don't have the same kind of pressure issues that we've got with the AV valves. It's just more or less gravity. There's a little bit of rebound pressure, but not a whole lot. So we don't have to have chordae tendineae. We don't have to have papillary muscles. Just the force of blood filling these concavities is sufficient to keep them closed. So we don't have all the extra stuff. We just have the valves themselves. And if we look at a fortuitous section, oblique section here through the heart, such that we capture all four of these valves, you can see kind of the arrangement of them, right? Such that here, looking from the top down, we have valves that will open this way. We can push those doors aside coming down, but not up, right? The AV valves here, the mitral, and the tricuspid valve there. And then you can see 1, 2, 3 versus 1, 2. And then the aortic and pulmonary valves here, concave up, here again on a real heart too, concave up, because again, they're trying to prevent blood from flowing down. Simple as that. And this is like my favorite photo in the entire book, honestly. This is a single papillary muscle here inside the right ventricle, so the tricuspid valve is here. Single papillary muscle with its chordae tendineae going up to one of the valves in that tricuspid valve. And you can see also here these trabeculae carneae in here, these ridges of muscle on the inside of that. Really pretty impressive stuff. And then again, a slightly different section here showing largely the same structures, right? Our tricuspid valve here, our mitral valve there, and our anchoring structures, chordae tendineae, papillary muscles, chordae tendineae, papillary muscles. Last thing we'll talk about today is what happens when valves go bad. Right? Like most things, they have the option of going shitty two ways. They can either be bad at their job and not stop blood from going backward. They can be incompetent. Right? I think we all know one or two incompetent people. Right? An incompetent valve is just bad at being a valve. Right? The issue with being incompetent, right, again, is the same issue we'd have if we didn't have valves at all. Blood flows backward. We're repeating effort, right? We're pumping the same blood again and again and again and again. We're reducing outbound pressure, so we reduce blood flow out of the heart. On top of that, not great, right, in the long run. The opposite of that is being actually too good at your job, right? So imagine, if you will, I don't know about you, when I was a kid in primary school, we had a hall monitor, right? It was usually a student. And every so often you get a hall monitor that was like just dead set on being like the authority, right? And you couldn't do anything without getting reported to your teacher. Well, this is what a stenotic valve is. It's an overly stiff valve. Rather than being too flimsy and letting blood just kind of go wherever the hell it wants to, a stenotic valve is going to really prevent blood from flowing where it's supposed to. It's not going to let as much through as it should. And so again, we have kind of the same issue. It requires more energy to pump blood than it ought to. Okay. The solution to this is a valvular replacement. We've kind of gone back and forth between using cadaver valves and pig valves. Pig valves, I think, were the first valvular transplant. And mechanical valves. I think we've kind of not perfected, but certainly gotten better at making mechanical valves than we used to. And so they're kind of the valve of choice right now. But again, it's kind of cycles, right? Typically with a cadaver heart, we'd like to transplant the entire heart because there's just not so many of them. Do consider donating your organs, please. And so that was really kind of the impetus behind using pig valves. They're roughly the same size as our valves. Pig organ transplant's actually pretty compatible with humans, just across the board. So as kind of the development of mechanical valves got better, we started using fewer and fewer organic valves, but you'll still see it done on occasion. Okay, so I will see you all tomorrow in lab. It'll be our last lab before the first lab exam. So come in and kind of be prepared to review.